So I will be very brief to just introduce that our next session coming up, a plenary session, is on understanding diseases, and it really epitomizes what the idea of the maps to mechanisms to medicine challenge is about. And it will be chaired by a member of the organizing committee, the Vice President of Research at Decode, and our friend Unnur þorsteinsdóttir. Unnur, over to you.
Thank you, Eric, and good day, everyone. In this session, we will go beyond DNA sequence variants for better understanding of disease. There are 3 speakers in the session, and each will have about 20-minute time slot for their presentation, including Q&A. The first speaker is Emma Lundberg. Emma is a professor in cell biology proteomics at KTH Royal Institute of Technology in Sweden and director of the Cell Atlas of the Human Protein Atlas program. She will talk about the spatiotemporal subcellular dissection of the human proteome. Go ahead, Emma.
Thank you so much for inviting me. I really enjoyed all the talks and particularly the discussions yesterday. And I feel like my talk doesn't really live up to the disease topic of this session. It more fits well in between the maps and mechanism, but I really hope it will be of interest and complement all the genetics talks that we've heard earlier. So I will focus on the aspect of measuring the subcellular partitioning of proteins and why I think that is an important aspect to understand cellular phenotypes. As you know, cellular processes are partitioned in time and space, and knowing where and when proteins are active or present gives us good clues about their function and the mechanisms they're involved in. And it is also established that protein relocalization is a form of non-genetic cellular regulation, and that's why I think it's so important to study this in addition to genetic studies of cells. As Unnur said, I'm one of the co-directors of the Human Protein Atlas project, and today I will mainly focus on the subcellular mapping, but of course it is a great resource with single cell resolved data with protein expression for the entire human body. So I hope that we can find good ways to leverage this rich resource also for ICDA, and would love to be involved in discussions about that. But what I do want to point out is that we use our protein-wide resource of antibodies to target and visualize proteins, basically. And initially, our work was very basic mapping. We created a subcellular map of the human proteome, just cataloging where are proteins, and we could localize the majority of the proteins that are expressed in cultivated cells, define organelle proteomes, very good information for doing basic cell biology studies. But today I will focus on some key findings that I think are relevant when we talk about understanding cellular phenotypes and understanding diseases. One is that 51% of all human proteins localize to more than one compartment. And this has also been shown for yeast and mouse. It seems to be around half. And these multilocalizing proteins, like you can see in this image, in green is the protein of interest, in red is a marker for microtubule. So the protein of interest here is both in the nucleus and on the plasma membrane of these cells at the same time. So these multilocalizing proteins may have a context-specific function and give rise to pleiotropic effects and even moonlight in different parts of the cell. So this greatly increases the functionality of the proteome and also the complexity of the cell from a systems perspective. And we've also shown that multilocalizing proteins are more prone to show differences in distributions, subcellular distribution between different cell types. So I do think that they are very, very important for fine-tuning cellular function. But when we talk about multilocalizing proteins, we kind of have to think about proteoforms as well, and then that's just, dauntingly complex. But to answer questions like how is dual localization achieved, are the protein population static or dynamic? We don't know the answers to this yet. What are the functional consequences? We don't know, but we likely need methods to study proteoforms, and that includes both post-translational modifications, but also coding variants, for example. What we do know is that 23% of the human proteome is estimated to have moonlighting activity. That's quite a lot. 78% of the known moonlighting proteins are involved in disease, and 48% are current drug targets. So these are important proteins. And also, just a number that blows my mind, the number of coexisting proteoforms in a cell is estimated to 6 million. It's just, I don't really know how to wrap my head around that and how to try to think about that. But that's just the baseline about the complexity of proteoforms in the cell that we know. So to give you one more specific example, this is one immunofluorescent image of genetically identical cells where we stained one protein with one antibody. If you're used to looking at microscope images, you can see that the protein is in some cells located to the nucleus, in other cells to the cytosol, in some cells both, and in other cells also to the plasma membrane. And then we also have this intensity difference. So there's also a great difference in expression level between these cells. And this happens to be a well-known moonlighting protein, so we know that the function is very different in these different compartments. So here, even though the cells are genetically identical, they display different functional capacities through this spatio-temporal partitioning in the cell, so to say. So we are interested in not only understanding this one protein, but understanding the entire proteome. What is the spatio-temporal heterogeneity that we see of the human proteome? So we turned to our large image collection in the HPA and searched for images where we see cell-to-cell variability, either that the protein is only expressed in a subset of the cells, or variation in expression level, or variation in spatial distribution. And in total, we can see that 18.5% of the human proteome shows cell-to-cell variability. And I should say that the number, if we look in the tissue atlas, is 17%. So it's very similar. So we believe that these variations that we see, cell-to-cell variations, reflect spatio-temporal proteome dynamics. But of course, we have static images, so we have to prove that. And there are different types of temporal dynamics, because besides spatial partitioning, partitioning in time is also a very relevant way for cells to partition different processes, so to say. And of course, you have short timescale variations like signaling or circadian rhythm or the cell cycle, and you can also have longer transgenerational variations. But for this, of course, we need temporally resolved assays or some kind of indicator to measure protein expression in relation to pseudotime. So we decided to start with the most easy task and understand how much of these cell-to-cell variations that we see of the human proteome correlate to cell cycle progression. And for this, we decided to use the Fuji cell marker as an indicator of cell cycle position, and we performed antibody staining and single-cell sequencing at the same time. And by the help of these Fuji markers, we could map the RNA and protein expression to a linear representation of cell cycle pseudotime. So if a protein shows cell-to-cell variability and is correlated to a cell cycle, we expect to see these cyclic patterns emerging, whereas if the variability is independent of the cell cycle, we expect a flat line. And this is exactly what we see. This is just some examples of well-known aniline, well-known cell cycle protein. We can see this temporal profile, determine the exact time of peak expression. Syntrine, a protein that shows very high variability, cell-to-cell variability, but not correlated to the cell cycle. And in every example, we had a negative control as well. So in the end, we could determine that these 1,564 proteins are all significantly variable. And we found around 25% of these proteins to be cell cycle dependent. And we find two groups of cell cycle dependent proteins here. The top one, represented by BAB1B and cyclin E1, are often well known. And here we see these typical cell cycle temporal patterns, both at the RNA and protein level. Of course, the protein peak of time expression is often lagging in the protein plots. Whereas for most of the newly discovered cell cycle proteins, we see a temporal profile at the protein level, such as for DUSP18 and DUSP19, that we could determine to be cell cycle regulators, whereas the RNA expression is stable. So we believe that these are regulated at a translational or post-translational level. So in total, to summarize, this is a very long study and it will be published soon, but there's a preprint available meanwhile. So 26% of the proteins that show cell-to-cell variability are correlated to the cell cycle. This is a much lower number than we expected. We found 300 new cell cycle proteins. Most are stably expressed at the RNA level. We found more cell cycle correlated proteins than we found cell cycle correlated genes or RNAs, which is also interesting. And there's many proteins of clinical interest here, also markers of cell proliferation. But I guess the point I want to make is that there's a lot happening at the post-translational regulatory levels when it comes to dynamic processes in the cell. So even for a well-studied process like the cell cycle, we can discover 300 new cell cycle proteins by doing single-cell spatial proteomics. But what is even more interesting is that the majority of the proteins that show cell-to-cell heterogeneity cannot be explained by cell cycling, which means that there must be other mechanisms, either deterministic or stochastic, that govern these variations. So here we decided to work with GFP biallelic tagging and live cell imaging, but this is ongoing work. I'm not going to show you too much, but this is a very striking plot. We can see that metabolic enzymes are greatly enriched here. So if we look at all human proteins, 18% show cell-to-cell variability. If we look at metabolic enzymes, 40% shows cell-to-cell variability of all metabolic enzymes. And this enrichment is true for all classes of metabolism. So this includes some cell cycle dependent variability, I should say, but the majority of the metabolic variability is not correlated to the cell cycle. I will just give you one example. This is an enzyme involved in cholesterol metabolism, HMG-CS1. Its function is in the cytosol. We can also see that it's localized to the nucleus where the function is unknown. So we see a spatial heterogeneity, but we also see this difference in intensity, which we believe is a temporal heterogeneity. So we've made a biallelic GFP tagged cell line, isolated a single clone, and then asked the question, can a single cell recapitulate this variability? Because that really means that it is temporal dynamics. And yes, it can. This is a GFP tagged cell line, and it's also different cell lines. So it shows that this heterogeneity is conserved across different cell lines. A single cell can reproduce all the distinct protein expression phenotypes. We still don't know what the function is and what the different functional capacities of these cells are, but we do know that it exists at least. And at this point, I usually get the question, but this is cell line, is it really relevant? Luckily, we have the HPA resource of images, tissue images as well. And here I can say that we see the same type of heterogeneity in many types of normal tissues and cancers, for example, glandular cells in stomach or acinar cells in pancreas, and where we see both the spatial and the temporal or intensity variations. And this is true for the 100 enzymes that we've tagged and tested so far. And I can also say that most of these variations seem to be at a shorter time scale than the cell cycle. So it might be that they actually correlate with cell cycling at some kind of higher harmonics level. We don't know that yet. But there's many questions to answer. What's the cause and consequence of these variations? What's the level of cellular regulation? Right now, everything is pointing to that this is mainly non-genetic, so post-translational regulation. Are there cellular states or intrinsic oscillatory systems we're yet unaware of? What's the function of these? We see many, many metabolic enzymes that go to the nucleus. Why? What do they do there? Is there a connection between metabolism, maybe drug metabolism, and some kind of genetic imprinting later on? We don't know. And how do metabolic and proliferative states interconnect? So this is something that we're working with in my lab now. But I guess what I want to say here is that I think that if we want to understand cellular phenotypes, it's important to complement genetic measurements with protein spatial measurements in order to get a complete picture of heterogeneity and not miss out on entire layers of cellular regulation, such as post-translational regulation. And for the last couple of minutes, I will shift gears a little bit and talk about how do we actually interpret spatial information and how can we integrate that with other types of measurements and what's the potential of doing this. So I just mentioned the localization of proteins in these images, and it's not trivial to deduce that. Even though it's easy by eye, it's not trivial to build machine learning models to do that because of many different aspects, morphological differences, multi-localizing proteins, mixed patterns, fast imbalance, and so on. So over the years, we worked with many different approaches, a citizen science, gamified approach, where we involved 300,000 gamers. We've done a Kaggle challenge. We're launching another Kaggle challenge later this year. So we're trying to engage a wider community here because we do have so many images that are a great benchmark data set to work with. But what I do want to point out is that we're finally at the point, if you look to the left here, where we have a deep learning model that we can use to extract features from any type of cell line. That we've tried 30 different cell lines so far. And this is where when we extract features from all proteins in the Human Protein Atlas, Cell Atlas, and just reduce the dimension and show it in the UMAP plot. And as you can see, we can distinguish all 30 different subcellular structures and substructures. But most importantly, the multi-localizing proteins, like D here, end up right in between the pure clusters of the respective pure patterns. So we can actually use this model to quantify information about mixed patterns as well. So we were finally at the point that we thought that now we can actually integrate spatial data with other types of data. So what do we want to do? So we teamed up with Trey Eidecker, who's an expert in building at UCSD at building hierarchical models of cells. And we decided to prove this concept for HEC cell line, where we imaged a set of proteins, 700 proteins using microscopy. So distances can measure distances of 200 nanometers to microns, and we embedded this information using our deep learning model. And then the Bioplex team at Harvard did their protein-protein interaction measurements. So that's like very close nanometer distances, and also embedded that information with the Node2Vec model. And then we can take these distances and translate it to pairwise protein distances and calibrate the physical distance in nanometer, which is pretty cool that you can calibrate networks to physical distances. And we could build this multi-scale subcellular hierarchy. But what is nice here is that by combining these two data types that are measured at, let's say, different resolution, we can find many new systems. So this model that we call MUSIC reveals 69 cellular systems, of which half are undocumented. That would be all the purple ones here. And we can validate some of them and reveal new roles for uncharacterized proteins and crosstalk between cytoplasmic and mitochondrial ribosomes, for example. So we believe that this type of model, the advantage is that we capture the pleiotropic effects of multi-localizing proteins and give protein interactions a spatial dimension. And we believe that this is one way of integrating data across scales to really leverage all the public data that is out there. So just to really show how much more information we get when we combine these two data types across scale, this is if we generate the same hierarchy with image data alone, we basically capture just a few systems, the nucleus, the cytosol, and the nucleoli. And if we only use the protein-protein interaction data, we capture around 12 systems, but when we integrate them both, we reveal 69 systems. So there's really synergy here. And with that, I would just like to end and give a little outlook. We work a lot with highly multiplexed imaging. That's definitely the future when it comes to protein imaging. So we can look at how the co-expression between variable proteins, and we want to identify the key drivers of non-genetic proteome heterogeneity. We're also working with generative modeling. It's a very cool project that I can talk about some other time, and to build a whole proteome subcellular map. And the aim here is to include genetic variants to start to explore spatial proteome perturbations. And also, big fan of open science. All our data is freely available through the HPA, and the new Kaggle challenge is on the way. And as a last slide, I thought I would just maybe summarize my thoughts in the maps to mechanism to medicine aspect. And this mainly goes in line with the recommendation 8 and 9. So sorry for the not lack of disease focus here. But I do believe that spatial proteomics can play a role in mapping cellular phenotypes and non-genetic cell heterogeneity and pleiotropic effects. I think we can provide missing links to understand cellular mechanisms, particularly when it comes to dynamic transition states that later give rise to genetically imprinted and inherited traits. And integration across of data across scales provide opportunities for data-driven discoveries of novel systems of importance for cellular mechanisms. And I also want to say that I think that there's, from the proteome perspective, there's still a great annotation bias for proteins, with a minority of the proteins having a majority of the research focus and having a majority of the annotations in all the different annotation databases. So I think that we need to be aware of this and avoid this inequality to allow discoveries of mechanisms that are not fully understood and maybe involves more uncharacterized proteins. And with that, thank you all for listening. It's great to be back in Sweden with my amazing team that you can see here at the top, and they all do such a great job. And I want to thank all my funders and you for listening. So happy to take any questions if there's time for it.
Yeah, we have some time. Thank you for a very interesting talk. So I have one question from Tuli. Do you have any insight into how differences in splicing, UTR, etc., at the RNA transcript level underlies the protein level variation?
Yes. So for the cell cycle, we can say that splicing accounts for 3% of the cell cycle dependence, for example. We could not show a lot there. The UTR we're currently looking into. That would be, I do believe that we will find some effects there, but I don't have a proper answer to it.
Okay,
but that's the next step.
Yeah. Okay, there's another question. Aisvarian Subramanian. That's the name. I'm not sure. I don't think I spelled it correctly. How you looked at the association of RNA localization with have you looked at the association of RNA localization with protein localization?
No, we have not. Would love to do that, but it would be very interesting to see if we can predict protein localization from RNA localization, for example.
Okay, there's room for at least one other question from anonymous individual. Did you look at the spatio-temporal patterns of cellular protein in organoids?
No, not yet. We're still at the point that we're looking at 800 metabolic enzymes. So when we know which the key drivers of this non-genetic heterogeneity are, then we will turn to better model systems like organoids. But so far, we're at least confirming that we're seeing the variability in tissue sections. So that's at this point.
Okay, then thank you very much, Emma, for a very good talk. And we have to move on to our next speaker, which is Nancy Y.I.P. Nancy is the Vice President of Research and Development, the Morningside Professor of Life Science, and the Director of the State Key Laboratory of Molecular Neuroscience in Hong Kong, University of Science and Technology. Her talk is titled Transcriptional Reprogramming Promotes Beneficial Microglial Function in Alzheimer's Disease. Go ahead, Nancy.
Thank you so much for the invitation. I'm very happy to have this opportunity to share with you some of the latest findings from my lab on microglial functions in Alzheimer's disease. As you know, Alzheimer's disease is the most common neurodegenerative disease, and yet there's no effective treatment. So the pathological changes include A-beta and tau, reduced synaptic remodeling, and neurodegeneration. Emerging evidence indicate that dysfunctions of microglia play an important role in impairing innate immunity. So for today's talk, I'd like to focus on our work to elucidate the molecular mechanisms that drive the functional changes of microglia in the pathogenesis of AD. And I'll focus my talk on a cytokine called interleukin-33. IL-33 is a member of interleukin-1 receptor family. It binds to a heterodimeric receptor complex, ST2 and IL-1 receptor accessory protein. It acts as an alarmin rapidly released upon cellular damage to maintain immune homeostasis. Now, increasing evidence suggests that there is a link of IL-33 and AD. Genetic variants of IL-33 and the receptor ST2 have been identified that are associated with AD. IL-33 expression is decreased in AD patient brains. So we're interested to know whether IL-33 signaling is involved in AD. Now, IL-33 act on its receptor complex to elicit cellular functions on target cells. ST2 also exists in a soluble form, and it can inhibit the functions of IL-33. I mentioned earlier that in AD brains, there is a decrease in IL-33 expression. And my lab also reported that in individuals with mild cognitive impairment, these individuals have a high risk to develop AD the level of soluble ST2 in the serum of these individuals is higher than that of normal control. We also recently showed that in AD patients, the level of soluble ST2 in the serum is also higher. Now, interestingly, plasma soluble ST2 level is negatively correlated with the cognitive performance as assessed by the MoCA score. And recently, we also found that in the CSF of AD patients, the soluble ST2 level is higher than that of healthy control. So we are interested to know whether IL-33 replenishment can ameliorate AD pathology. So we injected IL-33 into APP-PS1 mice intraperitoneally. And we found that IL-33 injection can alleviate memory dysfunction. It can restore the impaired hippocampal synaptic plasticity and also reduce A-beta deposition in APP-PS1 mice as shown in this quantitation here. So how does IL-33 bring about this beneficial effect? Well, since microglia plays a very important role in the clearance of A-beta, we investigated whether IL-33 can regulate the interaction between microglia and A-beta. Now, the first step is for us to cross the AD mouse model APP-PS1 with 6-3CR1 EYFP mice to generate a line that express EYFP in the microglia. So this is shown by the green color here. So these are microglia. And we also label A-beta by a chemical dye called CONNECT3, and is indicated by the red color here. Then we monitor, we track the movement of the GFP expressing microglia using 2-photon imaging. And under control situation, microglia remain rather stationary. But after IL-33 treatment, microglia will migrate towards the amyloid plaque. And this figure shows the images taken at 12 hours after IL-33 treatment. And you can see that the microglia is migrating towards the plaque. And so the mobility of microglia following IL-33 treatment is enhanced. And I should say that it is a subtype of microglia that elicit this increased mobility. Now, these microglia also seems to have enhanced ability to uptake A-beta, as shown by experiments using methoxyXO4, which can label A-beta, followed by flow cytometry analysis. So how does IL-33 bring about this state transition of microglia? So the first step we did was to undertake a global RNA sequencing analysis at 3 time points, 3 hours, 8 hours, and 24 hours after treatment with IL-33. Now we found that IL-33 treatment can bring about a stepwise transcriptomic programming in microglia. So including the early change at 3 hours, pathway analysis shows that this is associated with cell activation and stress response. And there's a subset of these genes that remain elevated at up to 8 hours. And pathway analysis reveal that they are associated with chemotaxis and migration. So these results therefore aligns with our observation in terms of functional characterization that IL-33 can stimulate chemotaxis followed by migration towards the plaque. Now, in order to further understand the molecular basis that mediate the action of IL-33, we undertook single-cell RNA sequencing analysis. The microglia clusters under control condition and also after IL-33 treatment, they are quite similar. And we observed that IL-33 can bring about an induction of MHC2 genes in the APPPS1 mice. It also can restore the homeostatic signature in these microglia. So we then look at whether microglia can interact with amyloid plaques. Now, since these microglia subtype, they express MHC2, and that can be used as a marker gene for the microglia. So we perform co-staining analysis with MHC2 antibody and also IBA1 antibody. And IBA1 is the marker for microglia. And you can see that after IL-33 treatment, MHC2 positive microglia, they actually associated with A-beta, as shown by the quantitation here, that following IL-33 treatment, there's an increase in MHC2 positive microglia associated with A-beta plaque. And furthermore, these MHC2 positive microglial cells, they have an enhanced ability to uptake A-beta, as shown by this quantitation. So far, I have shown you that IL-33 can trigger and a change in the functional state of microglia, initially from homeostatic state to chemotatic state, so that the microglia actually they migrate towards A-beta, and once it interacts with A-beta, then it exhibit an increase in phagocytosis and hence A-beta clearance activity. So our result shows that the transcriptal profiling actually aligns quite well with what we observed in terms of the functional characterization. That is, chemotaxis precedes that of phagocytosis. There's also a gradual increase in disease-associated microglia. Now, we are interested to know if this can be regulated by epigenetic remodeling. Now, we want to know whether IL-33 can regulate the remodeling of the epigenetic landscape, and this can include chromatin accessibility and transcription factor binding that results in the stepwise transcriptome reprogramming. So, for example, the chromatin accessibility landscape can be reviewed by doing a TAC-seq, whereas histone modification, transcription factor binding can be reviewed by ChIP-seq. So we use a TAC-seq to compare the chromatin accessibility landscape of MHC2 positive IL-33 responsive microglia and the MHC2 negative non-IL-33 responsive microglia. And as you can see here, that there is an increased chromatin accessibility at the promoter region of MHC2 genes. And we also perform motif analysis on these chromatin accessible region, and we found an represented PU1 motif, suggesting that the increased chromatin accessibility actually enhances the PU1 binding to these regions. We subsequently validated this finding by performing the PU1 ChIP-seq analysis, which clearly shows that IL-33 can also increase the PU1 binding at these promoter regions of MHC2 genes. So our epigenetic profiling highlights the essential role of PU1 in controlling IL-33 responsive microglia induction. Now, in order to further investigate the role of PU1 in controlling the microglia state transition, we ICV inject a small molecule that can inhibit PU1, and then we perform the transcriptome analysis. And our analysis show that this inhibitor, the PU1 inhibitor, can actually abolish the IL-33 stimulated transcriptome profiling and also the microglial state transition. And we subsequently also perform immunohistochemical analysis to show that in the presence of PU1 inhibitor, it can attenuate the IL-33 stimulated A-beta clearance in APP-PS1 mice. So all these data really highlight the crucial role of PU1 in controlling the microglial state transition and also the beneficial function in AD. So IL-33 can trigger this PU1 dependent transcriptome reprogramming in microglia, and it induces MHC2 genes and also induces the homeostatic signature genes, and resulting in enhanced chemotatic response towards A-beta and subsequent A-beta clearance. So we are interested to know how is IL-33 signaling regulated in AD patients. So I mentioned earlier in the talk that the level of soluble ST2 in the plasma of AD patients is actually increased. So we have data from this Chinese cohort. So we subsequently perform whole genome sequencing and GWAS analysis using this particular cohort. And we show that the regulation of soluble ST2 is actually contributed by genetic component, as I shown in this figure. That is, we identify genetic variants at this IL-1 receptor L1 locus, encoding soluble ST2 and ST2. And we found that these genetic variants are closely associated with plasma soluble ST2 level. So we conducted further analysis, meta-analysis, using our in-house data, that is the cohorts from AD patients in Hong Kong and also AD patients in the mainland, plus 3 cohorts with Caucasian population. And our top SNP actually has a very good association with the reduction in soluble ST2, and it also is associated with a decreased risk of AD with odds ratio of 0.7. So this particular soluble ST2 variant also show protective effect in terms of the endophenotype. So on the left panel, you can see that this particular SNP is associated with the delayed onset age of AD. So for individuals that carries 2 copies of this SNP, the onset of AD is delayed by 2.3 years, and the dementia-free probability is higher than individuals without this particular variant. Furthermore, it is associated with an improved cognitive performance as assessed by the MMSE score. So this particular genetic variant certainly seems to have a protective effect in individuals that carry the variant. So we went on to look at the postmortem AD brain from individuals that carry this particular variant. We looked at the A-beta pathology. We also looked at the interaction of A-beta and microglia. And finally, we perform single nucleus transcriptome profiling. Here is some of the data that we obtained. So for the postmortem brain obtained from the 80 patients that carry this particular soluble ST2 variant, the A-beta plaque area is reduced. Furthermore, we also observed an increase in the interaction of microglia with A-beta in the AD brain from the individuals that carry 2 copies of this soluble ST2 variant. Now, we set up the single nucleus sequencing platform in order to perform the transcriptomic analysis to investigate how the pathways are deregulated in AD patient carrying this particular variant. So this is just a schematic showing you, how the analysis is done. And initially, when we perform this analysis, we were able to actually show that there is cell type heterogeneity in human brain, and we also observed the proportion for these particular various cell types, which include astrocytes, endothelial cells, different types of neuronal cells, microglia, and oligodendrocytes. Now we examined the transcriptomic changes in the AD brain versus the normal control, and we found that each cell type actually exhibit distinctive transcriptomic changes in AD, and they are associated with different functional pathways. For example, for microglia, there is a reduction in type 2 cytokine response. For astrocytes, reduction in synaptic signaling, oligodendrocytes, reduction in myelination, endothelial cells, increase in angiogenesis. So,
sorry, Nancy, but I think your time is expiring. Like you're up to like 20 minutes. So
I'm on my last slide.
Okay, thank you.
Yeah, sorry. Yeah, so studies are ongoing to look at the function of the soluble ST2 variant on the molecular and cellular phenotype of microglia in AD. So just to summarize, under AD, with the accumulation of A-beta, it leads to a number of consequences, including impaired synapse and also A-beta formation and neuroinflammatory status in the brain. With IL-33, we believe that it triggers the chemotactic response and increase in phagocytosis of A-beta, and it has beneficial effect in the model that we have studied, and we believe that this suggests potential therapeutic use of IL-33 in AD treatment. So I think that's all I have, and I acknowledge all the students and postdocs who work on this project and my collaborators. And this is a picture of our campus in Clearwater Bay of Hong Kong. Thank you very much.
Thank you, Nancy, for an interesting talk. We have very little time for questions, but I have one for you. And it is like the association with Alzheimer that you talked about in the beginning. Was it with a common or rare variant, or do you know which one? Is it both, or?
So the soluble ST2 protected variant that I show you is a common variant. We can actually observe it in about 40% of individuals.
Well, I was talking about the IL-33 variant.
Oh, yes. We were able to identify it in our cohort. IL-33 variant and also the ST2 variant that I talked about.
Okay. So the IL-33, was it like an increased function or a decreased function of the IL-33 that associated with Alzheimer, or?
So it is a decrease in function.
Okay. Well, thank you very much. I think we need to go to the next speaker.
Okay, thank you.
Thank you. The last speaker in the session is Sten Linnarsson. Sten is a professor at the Karolinska Institute in Sweden. He will be talking about the molecular architecture of the mammalian nervous system. Go ahead, Sten.
Thank you. Thanks, and thanks so much for inviting me. I'm enjoying the sessions here. I'm going to give a talk that's solidly on the mapping end of the spectrum, and give a summary of our recent efforts to map the molecular transcriptomic composition of the nervous system. So let me start by talking a little bit about a paper that we published a couple of years ago now, where we basically tried to survey the entire nervous system of the mouse using single-cell RNA sequencing. This was work led by Amit Seisel and Hannah Hochgener in my lab. In this paper, what we did was we sampled from the nervous system in large regions, as shown here on the right, but in such a way that we didn't leave anything out. So we covered all of the brain, covered most of the peripheral nervous system. So we have the sensory, the sympathetic, and the enteric nervous systems. And so we're missing the parasympathetic and a few other minor regions. In this experiment, we performed fully unbiased sampling, so that at least in principle, we could discover everything that's there. Of course, that's also a matter of the number of cells. We had here about 500,000 cells in this analysis. And we performed a number of analyses here on the various cell types that make up the nervous system. And I'm going to just give you a quick overview. So first of all, if you look at the totality of the nervous system, we discovered using these methods and with the sampling and the number of cells that we had, we discovered about 270 clusters, which we might call cell types. And this is a dendrogram showing those cell types. So each leaf node here is a distinct, transcriptional distinct type of cell in the nervous system. And below the dendrogram, there are two bands of annotation. So the first band here shows the neurotransmitter phenotype for each neuronal type inferred from transcription of cellular transporters for these neurotransmitters. And the lower band shows the location of the cell in the nervous system. Now, if you look at this dendrogram, what you can see is that based on their transcriptional identities, the major split here is between neurons and non-neurons. So all the neurons are on one side and all the other cell types are on another side. And that's clearly a a functional split. It's a split that's driven by all those genes that are expressed in neurons to make them neurons. So synaptic genes and ion channels and so on and so forth. If we now go in and look among the various neuronal cell types, what we see is that the first split among neurons is between the central nervous system and the peripheral nervous system. And this again is driven by gene expression. Interestingly, if you go into this data, what you find is that it's driven mainly by expression of shared genes in the peripheral nervous system. And these genes correspond to a developmental ancestry. So what we're seeing here is that the distinction between the neural tube and the neural crest. So when the brain develops, it starts basically as a tube. And from the dorsal side of this tube arises the neural crest, which are cells that migrate out and generate, among other things, all of the neurons of the peripheral nervous system. And so when we are looking here in the adult nervous system, what we're seeing is that all the cells in the peripheral nervous system retain a memory of their developmental origin. If we go into the brain, to the CNS here, these colored subtrees here, they show sort of categories of cell types. And what you see is that by and large, they also represent developmental compartments. So one of the earliest events during brain development is the compartmentalization of the brain in anterior, posterior, and dorsoventral compartments. And so you can see here, for example, the forebrain on the left here, the midbrain here in the middle, and the hindbrain and spinal cord, each form subtrees, showing again that these cells remember where they came from. So it kind of shows the great importance of development in shaping not just the morphology of the brain, but also the molecular and presumably partly the functional identity of cells throughout life. Now, there are many interesting things you can do with such a data set. I'm going to show you one thing here. Diving in now to astrocytes, which are supporting cells of the nervous system. So it's been known for a long time, at least 100 years, that there are morphologically two very distinct kinds of astrocytes called fibrous and protoplasmic, shown here on the left. And so the distinction is between these sort of slender astrocytes with slender processes versus those with the bushy, short processes. And then it's also known that in certain regions, such as the cerebellum, there are specialized astrocytes. But we were surprised to find actually a number of molecularly very distinct astrocyte types. So in A here, I'm showing a dendrogram. So this is part of the dendrogram in the previous figure, but focusing now only on astrocytes and ependymal cells. So if we look at the astrocytes, you see that we find 7 distinct types of astrocytes. And of these, the Bergman glia were previously known. These are the specialized astrocytes of the cerebellum, but the others hadn't really been described. And they don't really correspond to the distinction between fibrous and protoplasmic. We find fibrous and protoplasmic markers in many of these clusters. So what they do correspond to, actually, if you look at the plots here, at the bottom here, you see there are 2 major kinds, so the telencephalic and the non-telencephalic. And what distinguishes them is that the telencephalic astrocytes are located exclusively in the telencephalon, and that's why we named them as such. Whereas the non-telencephalic types are located in all the regions behind the telencephalon. And this is a very clear distinction. In fact, if you look at the spatial distribution, there is a really sharp border between these 2 cell types that goes through the brain. And so that leads us to believe that these are developmentally specified. And indeed, if you look at transcription factors that are involved in patterning the brain early in development and conferring anterior-posterior spatial identity, what you see is that all of these telencephalic astrocyte types, they retain expression of FOXG1, which is a telencephalic patterning marker, whereas the non-telencephalic ones express mid and hindbrain markers such as engrailed and HOX. And so this, I think, strongly suggests that astrocytes, quite like neurons, are diverse and developmentally specified in these developmental compartments. So validating this, we stained the brain using markers of these major astrocyte types, and clearly you can see the spatial regionalization of astrocyte types. So at the bottom here is the data. This is RNA in situ hybridization in using a multiplexed method. It's a little bit hard to see. So on the top here is it's a summary based on thresholding of the raw data, where you can see that this incredibly sharp border between the telencephalic astrocytes, which express MFG8, versus the ones that are non-telencephalic, that are present in the diencephalon and the midbrain and the hindbrain, expressing angiotensinogen. And you have some olfactory types, and you have the Bergman glia and the cerebellum. So what of the fibrous and protoplasmic astrocytes? Well, it turns out that every astrocyte type that we will here classify as a distinct cell type, each one of them is biased towards either fibrous or protoplasmic phenotype, given indicated by GFAP, which is a marker of this subtype. So you can see that the telencephalic ones and non-telencephalic ones, and even the olfactory ones, each exist in a sort of a continuum between fibrous and protoplasmic. So what I think we're seeing here is that there are distinct types of astrocytes, but each of them is biased by the local environment to acquire either a fibrous or protoplasmic phenotype. Now, I want to switch now to development, and this is work led by Joelle Lomano in my lab and Kimberly Siletti, also in my lab, where we wanted to see what is the origin of this organization. Clearly, development has a great influence on the adult composition of the nervous system, but how does it arise? And we again wanted to do a kind of a survey of the complete process. So we dissected embryos at every embryonic day from E7 to E18. In the beginning, we took the whole embryo or the whole head, and then we gradually refined the dissections as the nervous system matures, dissecting forebrain, midbrain, hindbrain, and eventually distinguishing also ventral from dorsal regions. Now, with such a data set, you can start to ask questions about temporal aspects of development. So one interesting observation was that the total RNA content of the cell actually changes over time. So in early time points, you have a large population of cells that have a high content of RNA, so the total number of RNA molecules. This population gradually disappears as the brain matures. And we think this is largely driven by cell cycle. So if you plot here, here's a histogram of the expression of a set of known cell cycle genes. And the histogram is split by age. So for example, at E7, the black curve, you can see that the majority of cells express a high percentage of cell cycle genes. And this gradually drops over time. And at E16 here, the vast majority of cells now have very little expression of cell cycle genes. And this coincides with a drop in RNA content. You can also start to see how this is allocated to distinct classes of cells. And this really shows a neat, or gives us a neat summary of gene expression, or sorry, of brain development. So at the earliest time points, the cell types that we discover are dominated by primitive types, such as the early neural tube, and the germ layers, and the neural crest. But this gives way to radial glia, which are really the progenitors of the whole nervous system, and that are going to give rise to all of the neurons in the brain. They in turn differentiate into neuroblasts, this brown segment here in the middle of fetal development, which then differentiate into neurons. And in the end, the tissue is dominated by neurons. We stopped here at birth, so we only get a small fraction of glia, or immature glia that we call glioblasts here. Now, this data set is very complex. It's actually much more complex than the adult data set because you have basically the same level of complexity at the end, but then you have all the developmental history of every cell type. So in these plots here, I'm showing the clustering that we performed in 3 levels, where we define major classes of cells, as I showed in the previous slide, subclasses, and then individual clusters, so individual cell states. But this plot here combines aspects of time, of spatial gradients, of maturation, and of various lineages. So it's a very complex data set. So the challenge was how to visualize this in a way that makes sense and that you can start to understand it. And so in doing this, we were inspired by old maps, which cartographers have been excellent in finding ways to display multiple layers of information in a single static figure. And I really like especially these circular maps, where you can use the corners to show details or other aspects of the data. And you can overlay information in multiple layers, showing geographic features and political features and so on. And by the way, this is Stockholm, where I'm sitting right now. So this is the North Pole, and it's a map of the North Pole. One challenge is how to show time on such a static map. And there is the idea of the isochronic map. So this is an example of a map of how long it takes a letter to get to Venice in the 1500s. So these lines here are sort of locations that are all 1 week or 2 weeks or 3 weeks away from Venice by mail in the 1500s. So using these sources of inspiration, we created this map that shows in the middle all the cells in this experiment. It's about 300,000 cells, organized by similarity and colored by major classes. So here you have the primitive germ layers differentiating into the neuroepithelium, the neural crest, giving rise to the meninges. The rest of the neural tube maturing into radial glia, which gives rise to all of the different neurons that we have, and eventually to glioblasts that will differentiate into astrocytes and OPCs. Around this plot are all the 942 clusters. So these are the cell states that we defined, shown as a dendrogram that summarizes major kinds of cells. So for example, from here all the way down here are all the neurons. Up here, this segment here is the radial glia. So just by looking at this, you can see that it must be the case that neuronal identity is defined quite late. It's not pre-specified in the neuroepithelium or in the radial glia, because the complexity of the radial glia is already here much reduced relative to the neurons. In the corners, we're showing additional information. So, for example, key markers that allow us to identify excitatory and inhibitory neurons or radial glia. Time shown here, embryonic time from E7 to E18, showing that again, the development starts here and it's kind of progressing outwards according to these different lineages. Regions, showing interestingly, for example, that neurons are very regionalized. For example, here's a whole branch that consists solely of 4 brain excitatory neurons, whereas glia are mixed. So astrocytes and OPCs are quite mixed. We do see, however, that if we zoom into the glia, we can still see the distinction between the telencephalic and the non-telencephalic astrocytes here. Cell cycle, so you can see when as radial glia and other progenitors exit the cell cycle is when they start to differentiate into their mature cell types. Now, I have time for just one example of a finding in this from this work, which is about the regionalization and the patterning of the brain. So one of the earliest things that happens in the brain that gives it all the diversity is the establishment of these spatial domains, usually along the edges of the neural tube. So these are called the roof plate, the floor plate, and so on. There are many fanciful names for these so-called organizers. Organizers express particular transcription factors, and they secrete morphogens that induce the expression of other transcription factors in nearby tissue. So we took these organizers and we defined the morphogens that characterize them. In this case, here I'm showing known morphogens. So this is basically the floor plate is defined by the expression of sonic hedgehog, which is the ventralizing organizer in the brain, and so on. We were surprised to find that there is a much larger number of distinct organizer cell types. So, for example, the floor plate is really distinct in different regions of the brain. It's not just one floor plate. And that these actually do express a number of distinct morphogens. For example, BMP7 here is much more highly expressed in the midbrain than in the other regions. Finally, we took these these genes that were defined in our single cell data as being putatively spatially restricted, based on belonging to organizers and expressing morphogens that are known to organize brain development. Took these genes and we teamed up with Mats Nilsson's group in work by Elin Winsland in my group and Daniel and Christopher in Mats's group. And we performed in situ sequencing for a set of 119 genes defined in this way that were putatively spatially organized. We stained these on 20 sagittal sections through the brain. So we have a whole 3D or pseudo 3D stack here through the brain. I'm just showing you here one example of one slice out of this data set, showing the beautiful organization that we can reveal in this way, where you really can see the distinction between the floor plate and the roof plate. For example, here's the roof plate of the midbrain, here's the floor plate. You can see the sharp boundaries between spatial domains in the developing nervous system. And you can begin to see the really intricate mechanism that gives rise to the brain in all its complexity. This is work very much in progress, but I'm hoping that we can reintroduce the spatial information into this data set that was lost in the beginning when we do dissociated single cell RNA sequencing. And with that, I just want to mention that we like Emma too, we are great fans of open science, so we share all the data. We share the papers in form of preprints. You can find the adolescent and development data sets at mousebrain.org. You can download all the raw data, all the annotated data, and there are browsers where you can explore this data, including an interactive version of this sort of circular atlas. And yes, you can download different versions of the data here. The development data set is still in a preprint, so you can click on preprint to get the bioRxiv paper. And finally, big thanks to all the great people in my group who contributed, or our collaborators. I try to mention them along the way, our funders. And if you're interested in this, we have our webpage and mousebrain.org, where you can explore everything.
Well, thank you, Sten, for a wonderful talk. It is just fascinating to be able to track the transcriptional landscape during development. And I'm sure that there are some people that will tap into this source to identify potential important genes for a lot of neurological diseases. I'm going to see if there are any questions. No, there's nothing from the audience. The high RNA content in cells that are cycling, wasn't that, or in early development? And you were thinking that that was in relation to cycling.
Yes, and actually in this data set, it looks like RNA content starts high and then just drops. But we know if we compare with adult cells that adult neurons contain a lot of RNA. So there must be an increase that we are not capturing here because we're basically ending here at birth. But if we would follow these neurons along as they mature, we know that they will end up with a high content of RNA. So it's not a simple phenomenon. It's related to the identity of the cell and what it's doing and how much RNA it needs and the variety of that RNA. But there's a clear transition here from the earliest progenitors, which are very active and very full of RNA, but most of that RNA is related to cell cycle. versus neurons, which have a lot of RNA, but it's much more diverse and not related to cell cycle at all.
Have any comparable studies been done with human brains?
Yes, so there's been several studies that have included the brain or parts of the brain, for example, cerebellum, but so far not a comprehensive survey like this. We are engaged in projects to do that, and so it's coming, but we need a bit more time to finish that.
Sounds good. I think we have to wrap up, and I will transition over to Rachel that is gonna close the session.
Great, thank you so much. Thank you, Unnur, and to all the presenters for kicking us off with a really great session today.